And we have begun. So, well, that's done. This, I'm going to start with, even though this was the only class where I really got into it a little bit, so I want to redo it. Basically, just to go over the fine points. So there's really, it's interesting, these last two sections. This one has a lot to do, and we should get to the energy component today, which is important. Understanding where fatigue is, where waste products are. That's really probably the important part. Comes at the latter portion of this particular PowerPoint sequence, and then basically after that, there's not a whole lot. Then it was all about smooth muscles, which will conclude Monday. So because of that, I will send the next study guide. And I do not know what your exam schedules are vis-a-vis. This third exam will have to get in either. Typically you get bombarded during the last week before Thanksgiving break. I mean, I was tentatively eyeing Friday, and which would be 2 weeks from today, or the Monday of right after that, which would be, I guess, the 17th, because I think we're done, right? Something like that. Unless I miss my calendar, right? Because there's no school that week. Thank you. We'll know more as time comes on. So I talked about this a little bit, this whole idea between isometric and isotonic. Really, all you have to understand is that basically as we start to fire a muscle, it builds up tension. Those little ratcheting starts to occur. It doesn't translate to movement until we overcome the load. And we'll see this a little bit more. We start with just some isolated fibers until they achieve movement that we can visibly see. Typically, even if there's no load, if we're just doing this, still I had to initiate it with some degree of tension. So we always start isometrically. The length doesn't change. And then once it starts to shorten, then we typically maintain the same tension. It's a little bit different if we're looking at specific sports or activity. I mean, I would think it'd be different if you were hauling off and punching somebody, but I mean, that's not something those are, rare activities. And that's everything. We're looking at typical activities, the way things are. So it varies in response to the stimuli. And again, the thrust of this is to see, for the most part, frequency equals amplitude. It's a little bit different in muscles, as you will see. Once we get enough frequency of contractions, we can't make them anymore, as you will see. But it does play a role in how the ability of the muscle to relax. Very, very much so. And this is the whole idea of what's called a motor unit. It's basically where the nerve, or those axons of the nerve, target a specific muscle. Okay, and so this is really where we look at the functional unit of it. The definition is always, to me, a little bit hazy. So it consists of, again, depending on the nerve and all of the fibers. Again, the less fibers, we're able to do very intricate things, fine movements. Typically, imagine hands, facially, other parts of the body. Really, there are things that it's quite remarkable. Okay, and so it's more the fiber number, the greater degree of control, and these big, powerful muscles typically have considerably less. We saw this illustration before of actually defining what a motor unit is. We look at the variety of fibers and how many are serviced per se by this individual nerve. It's there. And so when we're going to talk about a twitch, and we're going to talk a little bit about why the importance of these myograms, because a fair amount of the students, okay, are in this, particularly in the health science area, of which there are many in our classes that share this class along with the pre-nursing as part of the pre-health career track. This is part of rehabilitation and those kinds of efforts that are there. So understand what a twitch is. It's just an isolated, and I hesitate to use the term movement, it's an isolated electrical event that we rarely see unless one of two things, unless we're doing it in a laboratory or in a setting where we're just trying to make the muscle twitch. Okay. And then again, this is where the rehabilitative aspect, the physical therapy aspect comes in, or it's something irregular. When someone has a muscle, all of a sudden they get a twitch out of it, that they have no control over it. That typically implies damage. Okay, there's some irregularity in the nervous system that's firing that muscle inappropriately. That's a whole different lecture when we start talking about, that's not basic anatomy and physiology. That's when we're dealing with exceptional types of things that are occurring. So we will look at this, and again, the name of the tracing is called an electromyogram. Nowadays, they're done with some degree of frequency. They're done diagnostically to determine if a nerve is trapped, making a diagnosis of carpal tunnel syndrome, making a diagnosis of sciatic nerve impairment, and part of the criteria that you would have deciding what type of treatment to go about using it. And so this is, and I know we went over a little bit of this stuff. I know because everybody, when you do it pre-exam, everybody's more focused on the exam, and it's not necessarily coming in and being sort of imprinted so you can start working at understanding it. So there is a period called a latent period. That is true for effectively every muscle, whether it's smooth, whether it is skeletal, whether it's cardiac, and even for nerves conversing with one another. There's a finite period of time, although it can be very, very small. We're talking fractions and fractions of a fraction of a second that are there, called a latent period. Then we see contraction, and then we see relaxation. So you'll see the graph very, and the contraction is all about this rapid area, these rapid depolarization events, and the relaxation is all about the repolarization events. Even though the mechanical events, the electrical events will come first. And inevitably, contraction is faster than relaxation. That's just the way the process works. It's there. And they're showing it to you here graphically. This is typically something we could demonstrate on an EMG in the laboratory. I happen to have the technology. We do this with our advanced physiology students. We don't have a lot of access to it for everybody to use it, unfortunately. But I mean, so it's something. Here's that latent period. So here's an isolated stimulus that will generate some degree of contraction, what we would call a twitch, and then it would relax. Although, as you can see, for the relaxation to reach down to the base level takes a time. This is 1/10 of a second here. So we're not talking about something that's necessarily in our eyes slow or visibly slow. It doesn't take that much time. But this is a laboratory thing, and again, you only see it abnormally if there's damage of some type. And so there's variations. Okay, we use our eyes all the time, rapid and brief. Calf muscles, large, fleshy, they're enormous. We don't have the luxury here of seeing a cadaver to actually see it or to understand it. The illustrations don't necessarily do it justice. It's almost like you have to hold it in your hand to be able to understand how massive some of these muscles, particularly in the lower extremity, are. So this is the classic example. The lateral rectus, moves your eye to the left. LR6, lateral rectus abducens nerve, one of the cranial nerves. Gastrocnemius, the big calf muscles. Those are power muscles in the sense they accelerate very rapidly. So when you're jumping to get something, okay, playing a sport, doing an activity where you have to really fire fast, or running someplace, sprinting, they're the ones that are involved. So quick, boom, boom. And now the soleus, again, is a postural muscle. We use it all the time. It takes a little while to gather its strength, and so too, it takes a little while to relax, but we're using it differently. That's the distinction. And so those are really good examples, and it would be examples if I hadn't even seen this textbook, knowing so much about the lower extremity anatomy that I would have picked. So relatively smooth, only in the lab or with neuromuscular problems. We grade the responses. We're not going to do muscle grading. That's something they would probably work with you, for many of you, when you're doing a physical assessment laboratory. That's something that the students, the physical assessment lab, particularly for the nursing students, I do not know what the program is for the health science students, typically would be this term of next semester. We start to do that. And that's very challenging, by the way, because there's a whole lot. There's a whole lot of things that you have to do, kind of boxes you have to check off mentally to make sure you're doing, and then how you assess them. Now, So if we are looking from a myographic standpoint, we can do a couple of things. We can change the frequency or we can change the strength. And graphically, they do a pretty good job of showing you it's not what you would necessarily think. Intuitively, we think, well, if we have a stronger signal, generate more power. No, it's about the frequency. The nuance in muscles, once all of those fibers are moving, we can't make any more of them move. We've maxed out, so to speak. So single stimulus results in a single contractile response. That's the twitch. Switches, it relaxes. Maximal tension from a single twitch. And notice how the stimuli are spaced. We're talking about one, if this is 100 milliseconds, that's 1/10 of a second. So if we're doing 10 of those, okay, if we're doing 10 of those in a second, it's going to contract, it's going to completely relax, do it all over again. So a second stimulus delivered after relaxation is complete does not produce summation. In other words, we can't make this become a more intense process involving this muscle, which you will see. So the whole idea of what they call temporal means over time, results if two stimuli are received by a muscle in rapid succession. They don't have the time to relax. What's going on? The calcium has not been pumped back. Okay? So as long as we have calcium sitting there inside the muscle fiber and we stimulate it again, more calcium is going to come out, obviously up to a point of limitation. So that additional calcium is released within the second stimulus will actually shorten it more. So this is a long-winded way. This is how we lift more, how we make more activity. How I lifted something very light. Now I got to lift something heavy. I got a whole bunch of stuff I got to lug in from the car. Okay. I'm doing those stupid leaps, which I'm going to have to do all weekend. Kind of get the idea. You're going to see what happens. So even though you'll get a certain amount of relaxation, and since it hasn't relaxed completely, the implication is there's calcium still out there. Now we're going to squeeze more calcium out. Boom. That's that next twitch. And so on and so on. The name we give to this is, imagine if we drew a little plateau. It's called staircasing. I'll get to why this is all relevant. So as now, we're maybe doing not 10 per second, we're doing 20. 2 in roughly every 10-second interval. Just using round numbers to make it easier to grasp. Okay. We're not done yet. What happens in the lab, or what happens in a physical therapy unit, we start to increase it more. If we increases it, we'll get the maximum. And if we call this summation, and here's the interesting name, it's called tetany. So if further increased stimulating frequency causes the muscle to progress to a sustained quivering contraction referred to as unfused or incomplete, probably a better word, tetanus. We name the disease tetanus for what it causes, a muscle spasm that cannot be released. Voila. Now, we've notched it up a little bit. Here I got 30 of these for every second. And so now we've reached max relatively quickly, and it kind of is, it's almost like the muscle can't sustain it, quivering. If you can imagine that, you almost can do it. So it's shaking. That's there. And then lastly, if we really max everything out, we can fuse it and undergo complete tetanus because contractions fuse into one smooth, sustained plateau, which will lead to fatigue. That's the magic word here. Voila. Now we're getting almost somewhere at least 50 and probably 100 of these every second. Why in heaven's name would we do something like this in a laboratory or a rehabilitative setting? And so you can actually do this. We hook people up machines and do this, not to torture them, but to rehabilitate them. Okay. This is a common technique that some of you in sport here may have had called electrical stimulation. It's been around forever. It's low voltage. It's not going to hurt anybody. We're not electrocuting them. Okay. So it's something that has been used, and like anything in medicine and science, it's in favor, then all of a sudden it's not in favor. We used to treat fractures one way. Now, we used to use things external. Then all of a sudden everything would be internal. Now we're going back to external again. It's all of this type of thing that there is sort of a fad. Okay. And so what happens is all of a sudden we explore old things, tweak them, and make them new things. That's the nature of trying to, literally build a better mousetrap. But without that long-winded explanation, here's what you do. So someone comes in with a muscle, oh my God, it's a spasm. I can't break it. It's like they've strained it and it goes into spasm. Scientifically, in physical therapy, we'll do this. We'll hook them up to a machine designed to keep them like this. We're going to try to induce fatigue. If we can induce fatigue, we can break the spasm. This is what we used to do when people would come in with back spasms. All the time in physical therapy. You set them down there, you slap a couple of pads on them for this low volt stimulation. You'd crank it up, okay, and that along with muscle relaxers and whatever other treatment, pain medicines, etc. All of these types of things would be done in an effort to break the spasm. If we're trying to strengthen a muscle that had weakened because of a previous surgery or a previous injury, we go backwards. We try to we gradually increase it up to this level by doing the staircasing technique. We're trying to build strength in the muscle. That's why I'm presenting it to you. Not that you're necessarily going to do it. Not I'm going to make a big deal to it. Literally on the next exam, I might have a question or two about that, period. But to understand why this is relevant, okay? So if the frequency increases, the tension reaches near maximum. And so that's really the take-home point from all of these things. So now what happens? That's frequency, why it's so important. In the muscle, we can reach it to a point where the muscle cannot contract any longer. And if we do it rapidly enough, it will reach it, pardon me, the way I'm speaking, reach it relatively briefly and be sustained there until we decide to relax it mechanically, until the decision is made to stop the stimulus, not from us, but by some arbitrary machine. Recruitment. I got to keep doing it more. So we have different kinds in the stimulus strength. If it's not strong enough to reach threshold, nothing's going to happen. If we reach threshold, okay, we might actually observe a contraction. But if we get to a maximum stimulus, we'll maintain maximal contractile force because we've engaged all the motor units. Kind of what it looks like. So here, and this is really where you see it. If we're below threshold, if the stimulus strength is insufficient to get any of these units to contract, we're not going to see it. Once we get the minimal amount, we'll see a very limited number. We'll increase it, but once we get to the maximum stimulus level, what that means, all of those units have been recruited. So we get maximum contraction. All that means is once we have the maximum stimulus, it doesn't matter how much we go over that maximum, the muscle's not going to do anything more than it's physiologically capable. That's why frequency is much more important than the size of the stimulus. There's got to be a stimulus to generate a contraction, but once we've hit the maximum stimulus, it's all about frequency, re-stimulate, re-stimulate, re-stimulate. And so the size principle is part of it. The smallest fibers first, larger fibers next, larger than largest. And again, it's all asynchronously. It's not all in one area. The fibers will be here, they'll be here, they'll be here to resist fatigue. That's by design. So it's asynchronously is a good. They're not all going at the same time. And they're just showing you small fiber motor units, then medium, then large. I don't get a lot out of that, but that's how it works. And this is something I mentioned before. The tone of the muscle. Muscle basically always has a degree of stimulation because of reflexes. So they're constant and slightly contracted effectively at all times. I'm not saying every one of them is stimulated simultaneously, because at some juncture, some have to relax and others are then stimulated. But for the most part, if we add it up, it's okay. I do that too. It's a long night. It's a very dull football game. So I had I'm leading my fantasy league from that game, thankfully. So, constant, slightly relaxed, slightly contracted, rather, state of muscles, spinal reflexes, alternatively, keep the muscles firm, healthy, and ready to respond. Part of something that's universal in the human body and universal in a lot of systems is called use it or lose it. How many people ask me, why are you still doing this, Lou? My mind is sharp. I wish my body was sharp. But I don't use that nearly as much as my wife will insist. So the whole idea, again, it reverts back to isotonic. Muscle changes in length and moves the load. And here's all you need to know about concentric and eccentric. Or if you want to call it eccentric, that's fine. Muscles are still exerting power, whether I'm contracting it, picking something up, putting it down. The whole idea is the muscle shortens to do work, or the muscle lengthens while we're generating force or doing work. That's the difference between concentric and eccentric or eccentric. Doesn't matter how that's how we were taught. We accented the E. Again, a lot of that is something, the way any of your professors, any of your instructors, whether it's here, it's in a clinical setting, wherever, however they learn to pronounce it or they learn to talk about it is how they will teach you. Doesn't necessarily mean it's universal. That's all they're saying. Don't care about that. Okay. Isometric, we can't make it short. We still do work. Okay, electrochemical, mechanical events, are they the same in isotonic or isometric? The results are different. In isotonic, we can shorten them. In isometric, it can't. We generate force, but we just cannot basically do anything about it. The filaments don't shorten. If you want to say spinning their wheels, that's fine. Don't care. So just as you know the distinction between isotonic and isometric, and the fact that every time we do something, it starts isotonic. I beg your pardon, it starts isometrically and then becomes isotonic. Because we have to first generate tension before we can move something. Pardon me for misspeaking. Finally, the whole idea on the energy. That's really the thrust of today. So I got through that. So to put it in perspective from an exam perspective for this next exam coming up, what do I really have to know about? Okay. Twitches are something we do in the lab. It's not something that happens ordinarily unless there's damage, neuromuscular damage, nerve or muscle not working properly. Okay. The big idea is frequency. That's the take-home point for this. Okay. So, and we do it over time. In other words, how frequently this idea of summating means if we stimulate it before it has come to a complete degree of relaxation, we can actually generate a greater degree of contraction. Okay, if we only allow it partially to relax. So if we stimulate it arbitrarily with a typical muscle 20 times a second, we'll get this. If we bang it up and go to 30 times a second, we get something called unfused tetanus. Both of those are, again, things we typically do in the laboratory or do in a rehabilitative sense. If we really ramp it up to at least 50 and all the way maybe to 100 times a second, we will generate what's called fused tetanus. This is desirable because we can effectively, over time and treatments, get muscles to fatigue. We can break muscle spasms. It's not as easy as it sounds. Okay. That's really what's happening. More about that. It's much less so about this. So it's less about the stimulus. Once we reach maximum, it's maximum. It doesn't matter how far up we go about it. We're just sort of wasting energy. Right? That's what I'm talking about for this whole thing. Concentric, eccentric. That's about it. You know the definitions of isotonic and isometric. Start isometrically, then proceed isotonically. And now, how do we get all the energy? We know the energy comes from ATP. It supplies the energy to move and detach the cross bridges, pump the calcium back, pump sodium out of the cell, bring potassium back in after excitation, contraction, coupling. Here you go. All the energy we have stored lasts 4 to 6 seconds. Think of what you do. Very rarely do you do an activity, utilize a muscle for that period of time. It's extraordinarily rare. We almost do everything. This is all part of what is called anaerobic exercise. We have energy that's stored. We're not having to convert to a unique process we call aerobic exercise. Okay, everything is short bursts. A lot of sports are short bursts. Oh, tennis players are very fit. They don't do anything aerobically. It's the people who probably, because the cross country team is traveling today, and some of them are missing from the room. Guess what? They're the ones who are doing aerobic activity. My son with his marathoning, that's aerobic activity. There's no way he could have run that without being into aerobics. That's what you're going to see here today. That's why the energy part of this nugget of this particular PowerPoint coming at the end of it is really the critical piece. It's the best way to frame this. So what happens? The energy does all of these things. Yeah, we heard about the cross bridges. We heard about pumping it back into. We know those sodium and potassium pumps. They're busy little beavers that are there having a good time. So the available stores of ATP are depleted in 4 to 6 seconds, but we rarely use anything. I just stopped there. How long did that take? A second or two? For me to move, for me to gesture, for me to try to do something. Heaven forbid I would get up, but I'm too lazy today. I have to mentally prepare myself for going to the casino this evening. It's important. I don't do anything. I gave up the whole counting the cards and all that. When I was young, I could do that stuff. So the available stores do not get into that habit. It's very expensive and painful. So ATP is the only energy source for contractile activities. Therefore, we have to regenerate it. How do we do that? Next, 3 different ways. We use it up. One is called direct phosphorylation of ADP by creatine phosphate. ATP, you saw that very first area of presentation, those basic nucleotides, what this magical molecule, we have high energy bonds. Effectively, that first 4 to 6 seconds, we lopped off one of those high energy bonds. Where are we going to replace it? Not so easy. So we do have some stored. It's a magical substance called creatine phosphate. Now we get to the discussion about protein powders and things like that people are prone to use. I'm fine with it. You're young, you're healthy. If you're going to do something like that because you want to build up your muscle and do that's, hey, your body, your choice. Do what the heck you want. So they eat protein powders, and for the longest time they were eating creatine or creatinine powders, which is the derivative substance. Your body, the analogy is like a recipe. If you have enough of the building blocks, you can continue to make stuff. Okay, you're limited. How many chocolate chip cookies can you make unless you have more of those wonderful Nestle semi-sweet morsels in the Toll House recipe? You got to have enough of those things. If you run out, you got to make a different cookie. So those protein powders, your body metabolizes them. They don't magically make them into muscle. They break them down to amino acids. The amino acids then are the building blocks, and we can make more muscle. If you're doing that with an exercise program under the guidance of a trainer, your kidney can get rid of all the nitrogen that brings in. You'll be fine. I have a stepson who did that. Oh, it's a leg day at work. I mean, just a physical specimen. Some people got that. I mean, gym every day. That's fine. You do what you want to do. What effectively it does vis-a-vis your energy levels and your ability to do things, it makes more of this creatine phosphate. It's very, very helpful. We have a limited amount of that, as you will see. Then at that point, we have no choice but to go anaerobic, full-time anaerobic activity, meaning they're using those muscles so vigorously, oxygen can't get in. And we're going to start creating waste products, particularly one called lactic acid. Okay? Because the only way we can make energy, and this is not something that's part of this course, it's something the students learn in our cell and molecular biology course when they're really understanding the nuances of what oxygen does. What oxygen does is it allows sugars to enter the mitochondria, because oxygen is there to gather all those electrons. Remember, I told you this. Our energy comes from taking substances we have ingested or stored and chopping them up and basically sucking the energy out of their electrons. Trouble is, we got to find a parking place for the electrons. That's oxygen's job. Since we don't have, when the muscles are contracting, we can't replace the oxygen. They're contracting, there's no blood flow. So here's how it goes. So we have a limited amount. We have another maybe 15 seconds to take a high energy phosphate from creatine. It's a unique molecule. It's in the muscle to start with. It can give up, all right, I can give you a high energy phosphate to make another unit of ATP. If you really got to get going, if you're in that sprint. Okay. Creatine kinase is an enzyme that carries out the transfer. The muscles have enough of this ATP and on top of it, the creatine phosphate reserves for a total of about 15 seconds. We added another 10 seconds. Lucky us. So that 100-meter, got it. You see the way I'm driving at? It's rare that we do these types of things. It's important, believe me. And I just don't guess that any of you here are parents. Maybe you are, no big deal. But they would have to be little ones. Don't look at me like that. Remember, I told you how old my daughter was when she had her daughter. 15 and a half, not quite 16 years old. And that little girl start running out into the street, you're chasing after them big time. You go into panic mode. That's the emergency supply. Because yelling is not enough. So grab the kid before they do something dumb. So voila, all it does is it's giving up. You've created a lot of ATP, ADP rather, with a big D for dog in there. And now you're just making some more ATP, and I'm going to sap it out of my creatine molecule, my creatine phosphate molecule. So, I mean, at best, combination is worth about 15 seconds. Now what do I do? The dumb kid took off chasing the dog that took off. Our dog would be like that. That's why we're not going to be home, because he would go freaking nuts, and he's 75 pounds jumping up. You want to scare the kids? Don't look at me like that. My wife, very fetching. She used to dress in black with a witch's cap and go out and play as Candy. But she was more Glenda the Good Witch than. It's a great story. Did you know that Margaret Hamilton, the late Margaret Hamilton, who played the Wicked Witch, she was a kindergarten teacher. It was in Hollywood. They didn't make a lot of money back then in the 1930s. Okay? They were all contract players. She's teaching kindergarten to supplement her income. Her agent called her, said, Maggie, they want you to be in Wizard of Oz. She goes, what part? The witch. The agent's response, they want you to be the Wicked Witch. Oh, okay. Did you know she went on Mr. Rogers? Do you remember who Mr. Rogers? I know you do. She went on Mr. Rogers' show to talk about witches around Halloween time. And came in and explained what she was and all that type of stuff. She was just an actress. Never, it's all right. One day I'll tell you the Mr. Rogers story if you're nice. So, you didn't hear that one, right? So 2 ATPs are generated for each glucose that's broken down. We do that. So ATP can be generated by breaking down whatever we have stored in glucose. It's a process called glycolysis. Remember, I told you the only place where we have stored sugars is in muscle or our liver. That's your emergency supply. Whatever that emergency is. I'm using the analogy of running after a kid that's taken off an animal is taken off, you're fearful. Something bad's going to happen. There's an emergency, you have to race after something. You don't require oxygen. You can't get it because the muscle's contracting anyway. The glucose, don't have to go, this is part of what's called metabolic scheme. Okay, and we can generate a limited amount of energy from this. That's why we can't just use glucose alone. We have to have the oxygen. I mean, this is, if you didn't learn about this stuff, when you have oxygen, it can get into a mitochondria. For every unit of glucose, we make about 34 to 36 ATPs. If we don't have the oxygen, we make 2. And on top of that, we make a horrible waste product called lactic acid. And that lactic acid will go into your bloodstream, and eventually bad things are going to happen. So low oxygen levels prevent that from being able to enter the mitochondria and what's called aerobic respiration. So that particular anaerobic pathway, okay, what happens, it can't get into mitochondria without going at the muscles, compress the blood vessels. We can't get enough oxygen. The accident of oxygen, we call it anaerobic glycolysis, splitting the sugar. Just know it's an anaerobic pathway. And the only place we can park those electrons creates this thing called lactic acid. If you go into metabolic scheme, that's the kind of stuff you learn about. That would be a bio majors type of thing. That they would be confused by it. I'm not expecting anybody here to. If you want to know that, I'll be happy to try to explain it to you. It's very, very interesting. Truth be told, most doctors wouldn't know it. They learned it when they were in medical school, all about that stuff. And as soon as they got into their specialty, they forgot all about it. Trust me, that's what they do. Nobody do that. It's not going to help them treat their patients, really, unless they're in a specialty that does that. So glycolysis and lactic acid gets into the bloodstream. It's one of the things that happens. We can use it as an alternative fuel source for a limited amount of time, but sooner or later, it's going to give up the ghost, and you're going to have to stop, because the accumulation of that, we're going to have to eventually replenish it in such a way that it will convert back to sugar. That's done in the liver. So a much more limited amount of ATP, roughly 1/20. That's why I said 2 versus 30 to 32 ballpark. It produces a lot faster. That's so we can function. Look what happens. Because of all that, what I'm blabbing about here, that isn't a big deal. You're lucky if you get another 30 to 40 seconds. Okay. Longer race. That's about it. That's why you train sprinters differently, hurdlers differently than when you train distance runners. You're basically, these days, if you were training even somebody who were doing milers or 2 mile, and we didn't have the meters today. So if they were doing a 1,500 or doing a 3,000 or a 5,000 meter, but it was mile, 2 mile, okay. And then the cross country was longer than that. I was still trying to throw a shot put and couldn't do very good. I wasn't that strong. So glucose, glycolysis, ATPs, lots of lactic acid, big trouble. Sooner or later, there's a point of no return. Here's what you have to do when you finally have run out of those resources. So for me, the analogy that I would give you, if you're on a treadmill, which is what I do. I do cardio. You get on the treadmill and you start to get busy. Okay? You start anaerobically. Whatever activity you're doing, you reach a point, well, I'm going to do a workout here. It's horrible weather outside. I'm going to be in here. You're going to set it, whatever elevation you're going to set it at. You're going to set it at a speed, and you're going to be able to get comfortable. Maybe you'll do that for X amount of time, and you'll have it, and you'll be able to stop it, break, start up again, whatever the interval training that you're going to do is. In order for you to accomplish that, or for somebody like me, I have an idea of what speed I have to go. I'm going to be between 3 and 4 seconds walking, because I'm not running. Okay, and whatever elevation, let's say I'm 3, 4, 5, whatever the elevation is, and anybody can do, because you work your way up to that, and you change over into what is called aerobic respiration. That's marrying the use of your muscles, your heart rate, and your breathing. That's what it does. Because if you're using your muscles too much, the oxygen can't get in. It's compressing the blood. But you can scientifically increase that ability by tailoring your heart rate and your respiration. That's why they call it CPR, cardiopulmonary respiration. Okay? You have the two are married together forever. Okay? If you have to bring in 5 liters or 10 liters, however much air you have to bring in per unit of time to satisfy your heart, that's what you do. So you marry your breathing, and not only you're bringing in oxygen, you're exhaling your waste product, carbon dioxide. That's part of the deal as well. And so if you know this better than I would, you're younger, you're healthier, where all of a sudden you can see, you're huffing and puffing, but if you're doing it scientifically, aerobically, even I, who's not in great shape, but not terrible for somebody 74 years old, can get on the treadmill and do 2-3 miles. Okay? Not like I used to, but more. Trust me, I didn't use one of those things till I was 55 years old. Gym's perspiration. Ugh. I'm better now. Honestly, I went to this one health club that I first started and said, "Do you want to see the locker room?" I said, "Must I? I don't even like them. We got to change somewhere I know." And it wasn't that nice anyway. Produce 95% of your ATP during rest and light to moderate exercise. Nothing wrong with moderate exercise. Theoretically, your immune system, every day you should do about moderate exercise of roughly for about half an hour or so. If you do that 6 days a week, that keeps you, it's just the whole thing. What are you doing? You're moving all those wonderful waste products around. You're stimulating your bone marrow. It's making more white blood cells. It's making more antibodies. They're all out there. You're almost cleansing it and recycling it. You're using those muscles the same way anything else. It's all about that. Roughly the first 30 minutes is that. After about that, then you can start to get into, probably after that, you're really helping your heart. And then, so roughly at around that 30-minute, if you're doing it for the heart, 3 to 5 times a week, maybe more if you want to. There's no limitation on that. Then all of a sudden it'll transfer the longer, then you get that fat burn, which a lot of people want. You can do that. I'm not an exercise physiologist. That's why if you're going to do that, or if you know an older relative is trying to do that, they need someone to advise them. Go to someplace where they have somebody who's reliable and can give them a trainer. So slower than the anaerobic pathway consists of a series of chemical reactions that occur in the mitochondria and require oxygen. Okay? Breaks glucose, larger amount. They're saying 32. There's a variation. Fuels used include glucose from glycogen stored in the muscle, glucose bloodborne, and then that's the big guy. After you use all those carbohydrates, the next source your body does is it breaks down fat. Okay? Because free fat, and they have more bang for your buck. That's the whole idea of the ketosis diet. At one point I weighed 237 back in 2018. I lost almost 60 pounds when we did strict keto. I got to go back on it again. 2 years ago, I was down about 20 pounds less, and I got to go back on it. And I eat carbs, and I love them. So many carbs, so little time. It's a curse. It's almost socioethnic. Bagels. So after that 30 minutes, that's when the fat burn kicks in. That's where this is all about. So it will help many of you. Fortunately, I'm looking at young, healthy people, thank goodness. And the culture today, we're a lot more focused on activity. And that's really a problem, what I emphasize a lot when we get into an AP2, though I'm not doing it in the next term, is to talk about the single biggest problem we have vis-a-vis activity is we're getting a lot of young people, kids younger than you, middle schoolers, because we're much more sedentary. We're stuck in front of computers, video games, eating improperly, blah, 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 blah. I mean, I'm pot calling, hello pot, this is kettle. Too many of us are overweight. And So a lot of people are getting type 2 diabetes. These are called maturity onset. The old diagnosis had to be at least 40 years old. We're getting teenagers, pre-teenagers develop. It's called metabolic syndrome or syndrome X. It's bad. So here's kind of what happened. If you do that, as you can see, I went. You can do this for hours. I can do it for hours, and I'm not in it. I'm not in any kind of shape. But I'll go back, and I used to do every Tuesday, Thursday, Saturday, and Sunday. Those were my days because I wasn't Monday, Wednesday, Friday teaching. So all of those things will occur. The energy system used during sports, aerobic endurance, the length of time a muscle contracts, light to moderate activity for just for a long period of time. Anaerobic threshold, the point at which your muscle metabolism converts to the anaerobic pathway. That very brief 15 seconds, and now you have to do anaerobic activity. You used the available ATP. You've used the available creatine phosphate. Now you better do something about it. That's basically the way the story works with all of this stuff. And so they're showing you to more graphically heal stored muscles first, creatine, so maybe you get 15 seconds. Now, that's the sprint. That's the miler. And with training, we have people who can do this for longer than 200s, 400s, whatever, 800s, etc. And the end of the exercise, and here for hours. What is the problem is this. There's a point where your muscles fatigue and they start to tell you, I can't do anymore. You just have to stop. I can't run anymore. I can't chase that kid. I can't chase the dog. He's going to have to come home on his own and hopefully doesn't get hit by a car. It's basically what, I mean, you can't physically do it anymore. You have reached a point of no return. It is less about the lactic acid. It is much more about ionic imbalances. Irregular charged particles disrupting the membrane of the muscle cell without getting scientific about it. Okay. All of these things, the amount of inorganic phosphate from the breakdowns, blah, 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 blah. Scientifically, we understand it, but those waste products and those electrolytes are problematic. A lot of people have problems with those. I can't tell how many patients I had over the years. What do I do about these night cramps and muscle spasms? It's electrolytic imbalances. It's hard to manage. And it's a bigger problem with things. You'll hear about things called restless leg syndrome. We don't know whether it's circulatory, whether it's waste products. There's nutritional approach. There's a lot of different approaches. When there's a lot of different approaches, that's code for not everything works. And unfortunately, those become significant management problems. So for years, we used different types of things to deal with that particular problem. So that's everything I can tell you. Lack of ATP is rarely a reason, except in stressed muscles. And so we call that catching your breath the oxygen dam. Lactic acid is reconverted, blah, blah, blah. They call it this stuff. I call it catching my breath or an oxygen dam. Leave it at that. See you. Thank you. Bye-bye. I'll save the Mr. Rogers story for when I have time.